Hello, everyone. So, just wanted to make a quick video going over some of the topics that are going to be covered in your first exam, which I know is pretty exciting, more so for me because grading exams and teaching on exam weeks is very fun. So, let's dive in. Just a quick PowerPoint slide. So, this exam is going to be focused primarily on evolution by natural selection and the processes surrounding this idea. Now, there's going to be a little bit with phylogenies and a little bit about genome evolution, but mostly going to be covering Darwin's theory of natural selection, as that is the key part of this first section of the course. Now, I gave you this quote in our very first lecture together, and Charles Darwin said, "Through time, species accumulate differences. As a result, descendants differ from their ancestors. In this way, new species arise from existing ones." And so we're going to use this quote, this framework set forth by Darwin to really dive into what you need to know for this exam. So the first thing we discussed was Hardy-Weinberg equilibrium. Now remember, when we're thinking about evolution, we have to think about populations because evolution simply does not act upon the individual. It has to act upon a physical population of organisms. Now, what evolution is really acting upon, now we're thinking of it as traits, but remember, for every trait, there's, canonically speaking, a underlying set of genes. Now, Hardy-Weinberg equilibrium, again, coming in after the fact of Darwin's ideas, attempts to predict genotype frequencies, and it does this with five key tenets having to be met. Now, Hardy-Weinberg is essentially like what we would kind of consider a null model, right? This is basically what is happening in a perfect world. Now, for Hardy-Weinberg to be a thing, we have to have these five key parts. No mutations must take place. No genes are transferred to or from other sources, i.e. no immigration or emigration. Random mating has to be occurring. The population size should be very large, and there should be no selection. So remember, Hardy-Weinberg, while in theory a great idea, clearly it's not a realistic one, right? We know natural selection is always working. We know mating is not always random, and so on and so forth. So, but what Hardy-Weinberg does try to do is predict the proportions of the genotypes in a population. So we can look at the frequencies here. But again, Hardy-Weinberg um, not super duper, relevant, but it does act as this idea of what's happening in the absence of evolution to the distribution of alleles in a population. Now we can write Hardy-Weinberg simply as p squared plus 2pq plus q squared is equal to 1, with 1 being a frequency of 100%. Now we're looking in this case, we have two distinct alleles, allele p and allele q, and in an idealized world, the frequency of P and Q should be equal to 1. So if I have two distinct alleles, well, the frequency of A plus the frequency of B has to equal 100%. Again, this is just looking at one gene with two alleles. Now, this equation gets significantly more complex if we have multiple genes and multiple alleles, but for the purposes of this class, we're just thinking about, again, one gene, one allele. So hypothetically, if we have P equals the large dominant allele for black coat color, so a black cat is BB, so capital B, or capital B, the heterozygote, and the homozygous recessive. And we can look at the frequency of alleles in the population. Has to be 0.3. I'm sorry, it doesn't have to be, but in this case, it's 36% for the genotype of big B, 48% for the heterozygote, and 16% for the recessive, little b. Now, for looking at the frequency of gametes, i.e. the frequency they carry a big B or little b, well, we simply have to take this 0.36%, I'm sorry, this 36%, I should say, and we have to divide this 48% in half because half the allele comes from big B and the other half comes from little b. So that's how we get the 0.24 or 24%. So the frequency of B, i.e. big B in this population, is 36 plus 24, giving us a grand total of 60. And we can do the same thing for little b. where it's 24% in the heterozygote, 16% in the double recessive, giving us a frequency of 40%. And you'll notice that we have 60 plus 40 gives us a grand total of 1 or a frequency of 100%. So this is how we apply Hardy-Weinberg equilibrium to a population. It's not going to be much more complex than that overall. Now, one aspect of Hardy-Weinberg that we simply generally don't meet is this idea of genetic drift. Now, genetic drift, um, typically what we're seeing is random change in the frequency of alleles by random chance alone. Now, this typically occurs most frequently in smaller populations, and so this really doesn't super duper apply commonly to Hardy-Weinberg, but remember it does kind of go in the face of what Hardy-Weinberg equilibrium stands for, i.e. we need large populations in Hardy-Weinberg. Now, the size of genetic drift, i.e. this random change in allele frequencies, is actually inversely related to population size. So the larger the population, the smaller the effect of genetic drift, and the smaller the population, the larger the effect of genetic drift. And genetic drift, again, is completely random, right? And it sort of just ties back to this idea of what alleles are getting into what gametes that end up having to fertilize or be fertilized in the next generation. So again, it is completely random. And with enough time, it can lead to the loss of alleles in isolated populations and leading to some other leading to uncommon alleles becoming much more vulnerable. Now, there are two distinct types of genetic drift that we're briefly going to discuss. Well, actually, just one, I should say, but I'll make mention of the founder effect because it is kind of an interesting one. And the founder effect is where you have a small population going to a brand new area, greatly reducing the gene pool size in this brand new area. So that's our founder effect. But what you should really be focusing on for your exam is this idea of a bottleneck effect, which is the dramatic reduction in population size due to some event. And it could be drought, disease, or any other natural forces. But the idea being is the way a bottleneck effect works is simply as this diagram points out. So if we have a glass bottle and it's got a bunch of beads in it, and we shake some of those beads out, as you guys probably know, the glass bottle simply doesn't allow very many beads out at a given time. So as you shake and shake and shake, only a small number come out the back end, which again is a drastic reduced population. And as you can see, it's drastically reduced in genotypic and phenotypic diversity as well. You can see from this, very simple example, we've lost all the red beads in this idea. And you'll notice that the next generation, while larger than the surviving individuals, again simply lacks overall genetic diversity in the essentially the ratio of genotypes and phenotypes is skewed because of this bottleneck effect. And ultimately, what we're seeing here is actually a loss of genetic variability. Very much like you would see with the founder effect, what we're seeing with the bottleneck effect is a net loss of genetic diversity due to drought, disease, or any other natural effect. We also see the same thing, just as a note, when we have artificial selection. We see a net loss of versus artificial selection, where the breeder, or again, in this case, it's just any human, doesn't really have to be a breeder, simply selects for desired characteristics. Whereas we have natural selection, where the environment really dictates which individuals produce the most offspring. And in contrast, artificial selection is different than natural selection. While they're doing similar processes, ultimately artificial is directed, whereas natural selection isn't directed in that sense. The nature's not going, I want this, like a breeder is going to. Nature's just saying, here's the environment, here's the traits that survive in the environment, thus those are likely to survive. Now, evolution and natural selection are essentially two sides of the same coin. Now, remember, evolution is simply how things change over time. And again, we're thinking about populations, the frequency of traits in populations. Now, natural selection is the force that drives evolutionary change. Now, for evolution and natural selection to do their things, you have to have three conditions that have to occur. First, there has to be variation among individuals within a population. If there is no variability, there's nothing for selection to act upon. Now, this variation among individuals has to result in differences in the number of offspring surviving in the next generation, which we would call fitness. And we'll get to that in just a moment. And finally, the variation must have a genetic basis. So I could technically be more fit if I went out and got all ripped and muscular at the gym, but I simply can't pass that all that work I did at the gym down to my next generation. So it might make me slightly more fit, from an evolutionary context, but again, it's not something I could pass down. So we need to have all these three key tenets met of evolutionary theory. Now, as I mentioned, natural selection and evolution simply are not the same. Natural selection is a process, and evolution shows us the historical record of change through time. Evolution is driven by natural selection, but they are mutually exclusive things. Selection does not have to lead to evolution, but evolution cannot occur without selection. So, again, two sides of the exact same coin, but not exactly the same. So let's have a hypothetical example here. And this is the example I used during lecture, where we look at pocket mice. Now you see pocket mice have two distinct populations. They have black and they have tan. Now you also see in their environment, they have two distinct environments. They have black volcanic rock and, tan sand. Now you'll notice that, well, just by glancing at these mice, we see the black ones fit in much more nicely on the black volcanic rock, whereas the tan ones do not. And on the sand, the black ones stick out like a sore thumb, whereas the tan ones do not. Now, the selection pressure here is not the actual environment. It's not the rock itself, but what's actually selecting for this is their predator. So in this case, we have a hypothetical predator, an owl, and it is acting as a selecting force. So picking out mice that only live on the black rock that are tan and vice versa. On the sand, they're picking out black ones. Why? Because the black mice are adapted to the black rock and the tan mice are adapted to the tan sand. So the environment gives these different conditions, and we have a selecting force here from the environment, again, a natural one, this predator coming through and picking out ones that aren't better adapted to their environment. Now, as you can see from the example, it's not a perfect scenario, right? There's clearly brown mice on this black rock, and in this case, there's no black mice on the sand. But the idea is evolution is essentially trying to weed, not again, it's not a directed way, but it's trying to essentially make this population of black mice be far more predominant on the rock than it would be for tan. So again, evolution, it's not directed, it's not trying to get the best organism. It's simply trying to have give the environment, and the environment selects for what's going to live and what's going to have offspring. Now that's natural selection. Artificial selection is humans choosing the traits that we like. And we're not necessarily choosing these traits because we want the organism to be the most fit, very much like natural selection does. So the example I gave in lecture is teosinte. So what we have here on the left-hand side, number A. And we have modern day corn, which is the relative of teosinte in D here on the right. And well, I guess you can see the dramatic effects of artificial selection here. It's pretty dramatic, right? Teosinte to corn. But what we're choosing here is traits that we like as individuals. We like big corn, juicy, potentially has more nutrients. This doesn't look too appetizing. So we're choosing traits that are good, but corn as we know it versus teosinte, well, they're not equally as fit. Teosinte is much more fit for its environment than corn is even for its artificial environment. So artificial selection does not choose the most fit organism. It simply chooses the traits that we like the best, that we see as being beneficial. Now, conversely, we can see another hypothetical example of experimental um, evolution in Drosophila melanogaster. So it's the common fruit fly. And this is the number of individuals on the y-axis and the bristle count number in Drosophila. Now, the data from this experiment basically has a hypothetical situation where the researchers were selecting for different amounts of bristle counts. So this was their initial population. They had, on average about 40 bristles on the abdomen of these fruit flies. So not the most exciting trait, but an easy trait to select for. And you'll notice there was a substantial amount of variability, 30 all the way up to 50. So there was variability to select upon. What the researchers did is they applied a artificial selection pressure, mimicking a natural selective pressure, but again, it was in an artificial setting to select for individuals with low bristle count and those with high bristle count, making those in the low population be more fit and making those in the high population more fit given their bristle count. Now you'll notice that the initial population, the distribution of traits completely shifts depending on what selection pressure they in turn applied. When they put a selection pressure for a higher bristle count, the mean bristle count jumped all the way up to close to 90. Whereas when they applied a selection pressure for a low bristle count, the mean dropped to close to 10. So you can see how evolution is working, right? We have a selecting force. It's selecting for traits that are beneficial in the given environment, causing a shift in the traits from this count to this count or this count to this count. So again, seeing a shift in the distribution of traits. And this is how natural selection ultimately works. Doesn't like work by selecting for crazy things. It just simply shifts the distribution of traits. And you definitely want to remember that going into your exam. Now, another example we gave of sort of a combination of natural and artificial selection, which is the peppered moths. Now, Biston betularia is the peppered moth, and they're found in Great Britain. Now, natural peppered moths look like this. They're black and white, so they're peppered, and they blend in pretty well with the trees and the rocks that they live on. Now, in the wild, there was a mutant, as you can see, this black individual here, and he kind of sticks out on this natural habitat. However, this example comes to us during the Industrial Revolution, where the combustion of coal in power plants led to a complete change in the essentially the coloration of the terrain that they were living on, shifting trees from looking more like this to shifting trees to looking more soot colored. Now, the idea here is that the environment was changing. And while in the natural setting, white moths were 99% of the population and black moths are only 1% of the population, in the shifting environment, which again was caused by humans, what we ended up seeing was a shift in the distribution. We were seeing 99% under industrial conditions were being black and 1% were being white. Well, why is that? It's not just the surface. It's also partially due to the fact that, well, black moths blend in better on dark surfaces, whereas white moths kind of stick out like a sore thumb. So the selection pressure was artificial, but there was a natural selection component here. So kind of a cool situation where humans induce a change in the environment, but natural selection kicks in, shifting the distribution, shifting from white in natural settings to black in industrial settings. So a pretty cool example of the pepper moths. Now, ultimately, evolution aims, and natural selection aims to produce individuals that are most fit for the environment. Now, fitness is a loaded word. Now, fitness simply is at its basic concept is simply the one that produces the greatest number of offspring. That's ultimately how you measure evolutionary success. Do you produce the most offspring? Yes or no? That defines how fit you are. Now, that being said, there are other things that go into fitness. Now, the capacity to get food and to attract mates, that is also an aspect of fitness. But at the end of the day, it does boil down to this idea of producing the most offspring. And those that are the best adapted for the environment are in the best position to produce the most offspring. So natural selection does, in theory, select for those that are best adapted and thus are able to reproduce and produce the most offspring for the next generation. So that's kind of how we loop all of this together. Now, what is what is sort of our evidence for evolution? Now, one key aspect of evolution that you definitely want to remember is homologous structures. Now, homologous structures are simply structures with with slightly different appearances and functions that all have derived from the same body part in a common ancestor. Now, all life has descended from a single cell organism, so we all have an ancestor, but certain lineages of animals share a more common or more recent ancestor. Now we can look at are mammals. So we have humans, cats, bats, horses, and porpoises, all different types of animals, but all living in different environments. Now we can look at the bones of the humerus, the radius, and essentially the hands of these individuals, and you'll notice that they have very similar structures, albeit slightly adapted for their environment. Now you can see this most clearly in the cat and the human, where we have the humerus, the radius, and in our case we have an ulna, but the cats have fused radius and ulnas. But you'll notice that humans have carpals and metacarpals and phalanges, and cats have similar structures as well. We see the same thing in bats. The bones are thinner, lighter, but again, same structures. We see it in a marine mammal, the porpoise. We have the humerus, the radius and ulna, and the carpals, metacarpals, and phalanges as well. And we see the same thing in the horse, right? Same structures. But what you'll notice is that while they have the same structures, they are all essentially derived from this because they're all derived from the same ancestor, because all mammals share one ancestor. Um, and interestingly enough, they adapted to their environment. So humans adapted to grabbing things, right? Cats adapted to walking on, bats adapted to flying, porpoises for swimming in the water, and horses for running in the um, on the open fields. So we have this adaptation to the environment that they live in, but ultimately they descended from the same common ancestor. Now, another line of evidence that we see for this as well is what we call pseudogenes, which are essentially fossilized genes. So if we all have the same ancestor, well, or if we have a recent ancestor, as we saw in this previous slide, well, there was an underlying genome of that ancestor as well. Now, as these organisms evolved and essentially, changed over time, well, there would be remnants of genes from their ancestor as well. Now, a gene for, say, I don't know, running really well might not be super beneficial if you're a bat, right? Because bats fly. But it's super beneficial in horses and beneficial in humans as well, in cats. So that's something that would be lost in bats, now still present, but lost its overall function, but present and active in humans and horses and cats. Now, this is what we call a pseudogene. It's a gene that's essentially from your ancestor, but it's no longer functional. You can also kind of think of it like a vestigial organ. Like, for instance, humans have tails, but they're no longer really functional, right? They're just little teeny tiny things that we call our tailbones. But the gene, again, just because a trait disappears, the gene doesn't simply vanish from the genome. That takes an extended period of time for that to happen. And another example I can give you is that ice fish actually have colorless blood. And ice fish simply are fish that live in the Antarctic seas where it's very, very cold. But it's kind of interesting. Their blood is colorless, so it doesn't contain hemoglobin. But ice fish DNA actually has a non functional hemoglobin gene. So what this tells us is that icefish's ancestor had to have had hemoglobin if it has a non-functional hemoglobin pseudo gene. So that's what our pseudogenes are. Now, evolution, again, it's all about the environment. And there's this really interesting phenomena in evolution, which we call convergent evolution, where as an organism experience environmental conditions, this might actually be very similar to other organisms found in very similar environments across the planet. So when we see this happening, this is called convergent evolution. So if we have a trait, such as what we see in cacti, for instance, where they have evolved great water use efficiency in the Gobi Desert, in the Sonora Desert, in the American Southwest, in the Sahara Desert, right? Great water efficiency. Well, this is an example of convergent evolution. They have a similar environment, so they need similar traits to survive and reproduce. Thus, they've evolved similar biological functions. Now, another example I can give you of this, which is probably a little bit more easier to understand, less so about just random cacti, is our marsupials and placental mammals. Now, marsupials are only found in Australia, whereas we don't find them anywhere else, obviously. Now, Australian marsupials actually resemble placental mammals that are found in other continents. And I can give you an example of this. So we have Australian marsupials and placental mammals found elsewhere. And you'll notice that for each of these organisms, well, we have a distinct ancestor that actually resembles it pretty closely. Whether it's a mole, whether it's a flying squirrel, whether it's an ocelot or a wolf, there is something that has evolved to fill a similar niche found in Australia versus, say, in mainland China or something like that. So there's definitely capacity to evolve things multiple times. And there's multiple examples from our lecture where we see traits evolving multiple times in very different places. Now, evolution, as Darwin talked about it, was gradual. So it falls into this idea of gradualism, where there's simply a accumulation of changes over time in a very slow manner. And so this is what Darwin thought, and this is what a lot of people thought for a very, very long time. But as we saw with Peter and Rosemary Grant, when they examined the finches on the Galapagos Islands, evolution can happen very rapidly. And this is what we call punctuated equilibrium, where we have very long periods of essentially no evolutionary change, followed by very, very rapid change. It's kind of this really sort of interesting dichotomy, right? Gradualism and punctuated equilibrium are not mutually exclusive. They can occur together. But sometimes the environment is so strong, you have these really rapid change. And the other thing I'll make mention is they're both at the ends of a continuum. So there can be kind of a gradient of how fast things are evolving. Some things evolve very rapidly, others evolve very slowly. It just depends, and you can switch between them depending on the environment you're in at any given time. So that's evolution. So let's talk a little bit about the species concept. Now, remember, the basics of the species concept come down to the biological species concepts, which has to take into account two distinct phenomena. The first being the distinctiveness of a species that occur together at a single location, so they occur sympatrically. And then second is the connection that exists among different populations belonging to the same species. So, population of humans living in Africa versus in North America, right? Same species, but distinctive enough that we live in two distinct areas of the world. Now, Ernest Mayr was essentially the father of the species concept. He defined species as groups of actually or potentially interbreeding natural populations which are reproductively isolated from other such groups. And that's the idea, reproductive isolation. You can't mate with one another and produce fertile offspring. And so what that means is, well, if I produce an offspring and it cannot go on to produce offspring on its own, well, we are two distinct species. Now, as we discussed, it's not the best definition, but it works pretty well for the vast majority of species out there. Now, how do we isolate two distinct species from one another? Well, as you recall, there are two distinct mechanisms, prezygotic and postzygotic isolation mechanisms. Now, postzygotic mechanisms are after the zygote is formed, so fertilization has occurred. And this is hybrid inviability or infertility, where inviability just means the hybrid dies or the embryos don't develop, or infertility, as we see with the mule here, where the hybrid is simply sterile and cannot produce offspring on its own. Now, there's multiple ways we can prevent reproduction from happening before a zygote forms, so an egg, fertilized egg, including ecological isolation, where species are occurring in the same area, but they occupy different habitats and thus don't encounter one another. So maybe lizards living on the ground and lizards living up in the tree. We have behavioral isolation, where species differ in their mating rituals. So birds are a great example of this. We have one bird species has a completely different call than another bird species, thus they cannot attract one another. Temporal isolation, where there's different species reproduce at different times of the year or at different times of the day. So you could produce one species is in the morning, the other is at night, or maybe one species is in winter and the other is in fall. So just being separated through through time. We can have mechanical isolation, where there are structural differences between species preventing mating. The most common way we see this is incompatibility between essentially sexual organs, so the penis and the vagina. If the penis and the vagina can't come together, well, mating and fertilization simply cannot occur. And then finally, we have the prevention of gamete fusion, where the gametes of one species, i.e. sperm or egg, simply cannot function properly with the gametes of another species. So you simply can't have a fertilization event, or the chromosomes don't line up, or any number of things preventing the fertilization happening. So these are all of our mechanisms that we typically see keeping species distinct. But speciation is a two-part process. So it's initially we have identical populations that have to diverge, and then reproductive isolation must evolve. Again, one of the mechanisms we just discussed to help maintain these differences. Now, gene flow can homogenize and erase any differences between two species that are separating. But speciation is also more likely to occur when there's a geographically isolated population or populations that are ecologically isolated as well. And so, again, if we have a species of birds, or we have a species of beetles, or we have a species of trees, they have to be at some way separated. And that's the best way we form a species barrier. So in this case, we have the beetles, they can't cross the river, or we have trees that simply can't cross a valley. Right? Really easy way to separate things through space and time. Now, this type of separation, which is what we call allopatric speciation, where we have geographically separated, or what we call allopatric, populations that appear likely to have evolved substantial differences leading to speciation. Now, why is that? Well, it's simply the environment, right? If you have two species, one's on an island, one's on the mainland, well, the island's going to be different than the mainland. So there's going to be different selection pressures, different environmental conditions leading to these two species. So geographic separation is by far the easiest type of speciation we will see in these populations. But clearly, we can have a speciation event within a single population in a single area, which is what we call sympatric speciation. And again, it doesn't have to be geographically separated. Now, the one common type that we discussed pretty in depth was what we call polyploidy. So where individuals have more than two sets of chromosomes. It's a really common thing we see in plants where changing the number of chromosomes you have makes you inviable with another plant species, thus reproductively isolating you. Now, the other way we can see this actually is basically by two plants or two organisms simply occupying different habitats, thus evolving into different lineages within the same location. So we can commonly see that. Now, the other thing we see commonly is what we call character displacement, where two reproductively isolated but ecologically similar species come into contact. So it's kind of the opposite of allopatric speciation, right? Where we have two species that are separated and then thus we bring it together. Now, natural selection is interesting. Now, natural selection for each species actually favors individuals that use resources that are not used by the other species. So essentially, natural selection favors the lack of competition because if you have to compete less, you're expending less energy, which means you're more fit, which means you use your resources better, which in turn means you're more likely to produce offspring and you're more likely to produce more offspring. And again, all this comes down to is greater fitness. And what natural selection does, it selects for trait differences in resource use that will thus increase in frequency over time and ultimately leading to the divergence of the species. So we'll give an example of this where we have two species. We're looking at the frequency of body size here, and they come into contact. Species one is in this pinkish color, and species two is in this goldish color. Now, natural selection says this intermediate body size, which they have this overlap, simply does not work. There's too much competition. So natural selection selects for the extremes, making those with larger body sizes or smaller body sizes better adapted for a given environment, thus leading to a shift in the frequency of the traits. So you see pink now is dark red, and it's very different distribution of traits. And the same thing with this goldish color, right? Complete shift in the frequency of traits. This is what natural selection does. And if you reinforce this, again, you have distinct species either forming or the potential to form, again, all due to the lack of competition here. So that's evolution. Now let's wrap up by just quickly talking about phylogeny. Now, phylogenies are a way to represent the evolutionary history of organisms. And so phylogenies, we have these tree tips, which we define as the species of interest, A, B, C, D, and E. And we have a couple things. So horizontal lines in this case are branches. We have branch points or nodes is where two branches come together. So as I'm circling here, or as indicated in yellow here or yellow here. And we have evolutionary time or relatedness passing from left to right. So the way we interpret a phylogenetic tree is that the shorter the branch length, the more closely related two species are. So in this case, species A is much more closely related to species B because this horizontal branch length is smaller than, say, if I compared species A to species C. Because again, all I have to do is look at the size of this branch length. Smaller branch length, more closely related species. Now, we can also look at what we call another part here, which we call a clade. Now, a clade can be broadly defined, but clades are just groups of closely related organisms. So A and B could be considered a clade, or A, B, and C could be considered a clade as well. And then the final thing I'd like to make mention, E is what we would call an outgroup. So it's a species that is essentially very distinct from a clade that we're trying to examine. So in this case, our clade is A, B, C, D, and E would be the outgroup to this clade. Now, phylogenies can depict either evolutionary time or simply relatedness. It just depends on what type of phylogeny you are looking at. And remember, we can look at a phylogeny such as this, and we can see this. We have gibbons, humans, chimps, gorillas, and orangutans. And the way we've drawn this version 1, version 2, and version 3 are identical. So just because I've rotated things slightly between these three trees, the evolutionary relationships are still the same, right? I still interpret humans and chimps being more closely related than humans and gorillas. And you see the same thing across all three trees. So the relationships don't change. I've just rotated things to make things easier or harder to read. Now, remember, we can also make our own phylogeny by simply looking at the presence and absence of traits. So in this case, we're looking at traits here in the far left column. I'm sorry, organisms in the far left column. So lampreys, sharks, salamanders, lizards, tigers, gorillas, and humans. And then traits in the columns here: jaws, lungs, amniotic membranes, hair, snow tails, and bipedal. Now, remember, ones in this case are the presence of said trait, and 0 is the absence of said trait. Now, the more similar two species are in an evolutionary concept, the more overlap in traits they will have. So we can look at this example where we have gorillas and humans, and we're looking at all these traits. Now, humans have all 7 traits. No, I'm sorry, 6 traits. And gorillas have only 5 traits. So, based upon these traits, we would say gorillas and humans are probably more closely related. Now, next up would be tigers, which have 4 of the 6 traits, and then lizards, which have 3 of the 6 traits, salamanders that have 2 of the traits, and sharks that have 1 of the traits, and lampreys, which have none of the traits. So, if we were to take this information, the similarity here, we could actually draw phylogeny from it. And we would say, again, based upon the sharing of traits, humans and gorillas are most closely related. And so we would draw a phylogeny like that. Next would be tigers to humans and gorillas, then lizards, then salamanders, then sharks, and then finally lampreys. And then the final thing we can do is overlay some evolutionary innovations along the way. Now, if you remember back from this table here, well, one of the innovations for humans was bipedalness, i.e., the capacity to walk on 2 legs. Well, you can see we can overlay that here. Now, an innovation for humans and gorillas that's shared by nothing else is actually tail loss. So humans and gorillas have no tail. Everything else has a tail. And so we've noted that here. And then we have the presence of hair, which is again not found in any of our lizards or sharks or lampreys, but it is found on tigers, gorillas, and humans. So that's how we can take trait information like this and directly make a phylogeny from it. And so our last 2 slides are actually just going to be from our most recent lecture, looking at genomes and genomic evolution. So the first genomic term that you should be familiar with is synteny, or the order of genes in a genome. Now, in this example, we have a number of hypothetical genes: red, blue, orange, so on and so forth. And in soybean and tobacco, they're in the same order in the genome on different chromosomes, but the same order. Thus, they have the same synteny of genes. And we also have our final thing, or sorry, our second to last thing, which is horizontal gene transfer, where genes simply hitchhike from one species to another. It's also been called lateral gene transfer. And so we talked about this in the capacity of microbes, where microbes can acquire a gene from the environment or from another species, but they're not acquiring it from their parent, right? Which is how we typically think of the transmission of traits, right? We think of usually vertical gene transfer, i.e., I got my traits from my 2 parents, whereas horizontal gene transfer is like, hey, I saw a bag of Doritos, I grabbed the genes on the Doritos and I stuck them in my genome, right? That's the idea of horizontal gene transfer. So kind of a funky thing, but a really important thing in biology, uh, especially microbiology. And then the final thing is these genome terms. And these are kind of important ones to remember going forward. Now, we have paralogs, orthologs, and homologs. Now, homologs encompass both paralogs and orthologs, but homologs simply refers to the same gene, either found within the same genome or between organisms. Now, paralogs are two genes within a single organism that have arisen from a duplication of a single gene in an ancestor. And orthologs reflects the conservation of a single gene from a common ancestor. So if I have a hemoglobin gene in a cat and a dog, well, those are orthologous genes. Or if I have two hemoglobin genes within a human, those are paralogous, those are paralog genes. And then if I want to say that the gene in hemoglobin gene in a human and the same genome, those would also be just called homologs, and a genome gene in a cat and a dog would also be considered homologs. So technically homologs is correct to describe both of them, but the most technically correct way is paralogs is within an organism and orthologs is between organisms. So that's going to end our review session for today. I hope that provided a little bit more concrete studying direction for you. What I went through in this exam study guide is exactly what's going to be covered on your exam. So, you don't have to be guessing what you're going to have to study. The topics that I covered, reading through the book, reading through the slides will provide you all the information that you really need to kind of prepare yourself for the exam. So if you have any questions, please do let me know. But otherwise, I hope you guys have a great day. Take care.